Before we dive into the process, there are two things that you should keep in mind. The first is the structure of the eukaryotic ribosome, which consists of two subunits, the 60S and the 40S subunit. The 60S is made up of proteins and three major ribosomal RNAs. These are 28S, 5.8S, and the 5S ribosomal RNA. On the other hand, the 40S subunit has the 18S ribosomal RNA as its major component. And this entire protein and RNA complex in eukaryotes is known as the 80S ribosome. And the processing of the mRNA and the synthesis of the polypeptides occurs at these so-called E, P, and A sites in these 80S ribosomes, and we discussed these sites earlier in the translation series. So I will draw out the ribosome like these dome-shaped structures, but always keep in mind that over 60% of the ribosome is made up of ribosomal RNAs. The second important point is the structure of the eukaryotic mRNA. The eukaryotic mRNA begins with a 5' UTR, which oftentimes contains secondary structures like hairpins and stem loops. The 5' UTR typically also has a ribosome binding site, which in eukaryotes is known as the Kozak sequence. This 5' UTR is followed by the coding sequence, which starts at the start codon, and this coding frame continues until a stop codon is encountered. And towards the end of the coding sequence, you have the 3' UTR, which is followed by a poly-A tail. The 5' end of most all eukaryotic mRNAs have a 5' modification. You can learn all about these modifications in the transcription videos. The links are down in the description as well as on the cards above. Now, these modifications at the 3' and the 5' end recruit some specific factors, or proteins that cause the mRNA to take a loop structure, such that the 5' and the 3' ends come close to each other. We touched on this a little bit when we were discussing the transcription in eukaryotes, and we'll see later in this video how these interactions are useful for the process of translation initiation. The initiation process can begin when the two subunits, the 60S and the 40S, are not in the 80S ribosome form. This could be either when ribosome is being recycled from a termination step or when a new subunit is being assembled. So the point is that the 60S subunit usually has to wait because the initiation of translation begins with the 40S subunit. And this is exactly what we saw in the prokaryotes where it started with the 30S subunit. By the way, I will point out some similarities and differences between prokaryote and eukaryotic translation initiation as we move along in this video. So I hope you've watched the previous videos. So the first factor to associate with the 40S small subunit is the eukaryotic initiation factor 1, which binds at the interface region between the E and the P site. And later in the process, one side of this interface becomes the E site and the other side becomes the P site. This EIF1 is similar to IF3 that we saw in the prokaryotic translation. On the other end of the 40S subunit, the A site is defined by the binding of EIF1A. And this factor is very similar to the initiation factor 1 in prokaryotes. The middle ground between these two factors is mostly the P site. Now, all these interactions that occur on the 40S subunit are mediated by a larger eukaryotic initiation factor known as EIF3. This protein is almost as big as the 40S subunit itself. The C-terminal end of this EIF3 is pretty close to the A site, and the N-terminal domain is located pretty close to the E site. Now, let's talk about the functionality of these factors. The EIF1A primarily blocks the tRNA binding to the A site. And one additional function that it serves, which we will see later, is that it also helps in the docking of the 60S subunit. The EIF1, on the other hand, prevents the 60S subunit from joining. And there's one more function that EIF1 serves, which is that it positions the mRNA at the 40S subunit. And this large initiation factor 3, as we said, is a scaffold protein, which interacts with most all initiation factors to regulate the overall 40S subunit interactions. The one interaction regarding EIF1-3 that I would like you to note is that the initiation factor 3 subunit J, also called EIF3J, which is part of the C-terminal domain, contacts the 40S subunit to define the mRNA entry channel. So after these three initiation factors assemble, the 40S subunit is ready for the next step. In this step, the initiator tRNA, which almost always carries the methionine, is bound by the initiation factor 2. and this EIF2 is attached to a GTP. This charged initiator tRNA is different from the prokaryotic tRNA in that there is no 4' modification done to the methionine. And the function of EIF2 is similar to the function of initiation factor 2 in prokaryotes, which also helps bind the initiator tRNA and helps it to recruit onto the 30S subunit. And for your note, this anti-codon on this activated tRNA must be UAC. If it is not obvious why, it'll become clear in the next step. So let's just quickly complete the sketch of this 40S complex where we have the 3 initiation factors bound. Now this initiator tRNA, which contains the methionine, is brought to the P site with the help of eukaryotic initiation factor 2. And this EIF2 interacts with both EIF1 and EIF1A. And this helps in the positioning of the initiator tRNA. Once eukaryotic initiation factor 2 has positioned, another initiation factor called EIF5 binds to EIF1 and EIF2 to increase the affinity of this complex. And on top of this, the eukaryotic initiation factor 5 also has a GTPase activating domain. So this is the complex now, where you have EIF1 at the E and P site transition, the initiator tRNA at the P site, and the EIF1A at the A site. So this 40S complex where all these initiation factors are bound along with the initiator tRNA, but there's no mRNA bound yet, is called the 43S pre-initiation complex. And once this 43S pre-initiation complex is formed and the interaction between IF2, EIF1, and EIF1A is stabilized, the EIF1 and EIF1A cause a structural change in the 40S subunit, such that the mRNA entry channel now opens up, which means that this mRNA is ready to be bound onto the 43S complex. But this mRNA has to undergo specific steps before it can get recruited. And these steps involve the interaction of the 5' and 3' end of the mRNA. We quickly glossed over this step earlier, but now let's get a bit concrete and look at the details. The eukaryotic mRNA has a 5' cap modification, which is recognized by a protein called eukaryotic initiation factor 4E. The mRNA on the other end has a poly-A tail, which is bound by stabilizing proteins known as poly-A binding protein C1. These poly-A binding proteins recruit initiation factor 4G. But EIF4G not only has affinity for poly-A binding proteins, it also has affinity towards the EIF4E, which is located at the 5' end. So in principle, no matter where it gets recruited, either at 5' end or 3' end, the two ends later will come together to form a loop regardless. But let's just say for the sake of simplicity that it gets recruited at the poly-A tail. The EIF4G then recruits EIF4A, which happens to be a dead box helicase. And now this mRNA, after binding the initiation factors at the 5' end and the 3' end, can form a loop, such that the initiation factor 4E and 4G are brought together due to the capping and the poly-A tail modifications on the mRNA. The eukaryotic initiation factor 4A, which is the helicase, by itself has very little activation energy. So to help this factor, another protein is required, which is called initiation factor 4B. And simply put, EIF4B enhances the EIF4A helicase activity. Now this complex of initiation factors bound to the mRNA become active, and as a result, the helicase EIF4A moves forward by consuming ATP as its energy source. And when it moves, it unwinds the secondary structure, like hairpins and stem loops, in the 5' UTR. And this unwinding may be required to make the ribosome binding site more accessible. So if we sketch out the resulting structure, we see that the helicase has moved forward and displaced the secondary structure. So important thing to keep in mind here is the role of helicase in breaking the 5' UTR hairpins. To make things easier for remembering all this, note that initiation factors that bind to the mRNA are named 4E, 4G, 4A, and 4B. Essentially, this is saying that initiation factors in class 4 are involved in the mRNA binding, and after binding, they help in the loading of the mRNA onto the 43S pre-initiation complex. Now that this mRNA is ready for loading, let's see how this structure would look after loading. I'm going to draw out the 43S pre-initiation complex first, and while I complete this sketch, you can go ahead and subscribe to the channel if you haven't already, so you don't miss out on the upcoming videos. Don't be shy, subscribing is totally free. Alright, now for loading of the mRNA onto the ribosome, the EIF4 complex on the mRNA is recruited near the A site on the small subunit. So if you draw out this complex EIF4 on the 40S subunit, you notice that EIF4A is closely associated to the EIF1 near the E site. And the mRNA is threaded through this complex into the mRNA entry channel and looped out such that the 3' end and the 5' end are physically in contact, as we have seen already. Note that we said the initiator has the anticodon UAC, which means that its complementary sequence is the AUG, which is the start codon on the mRNA. But the helicase activity of EIF4A was simply done to unwind the hairpin so that it can bind the ribosome, which means the ribosome now has to find the start codon and then position it to the P site. So the 40S subunit essentially begins scanning the mRNA for a start codon, and this process is known as the ribosome scanning, which occurs in the 5' to 3' direction. This 43S pre-initiation complex now, after binding the mRNA, assumes an open conformation because the mRNA entry channel is open, because the ribosome now has to scan the mRNA. And this scanning process to find the AUG consumes energy in the form of ATP. The open complex of 43S transitions to the next stage when it finds this AUG. And let's just draw out the next stage. This will take me a quick minute. Let's just tweak this here and some lines and add some colors. And we're all done. So the change to notice here is that the AUG is now paired at the P site with the initiator tRNA. The EIF4A and 4B remain bound near the E site, and the EIF4G and 4E move out of this complex but are still associated together, which allows the mRNA to be in a loop. Now, there could be many AUGs, right? So how do you know which AUG should be positioned at the P site? It turns out that this positioning happens in the context of a sequence, which usually is this G and C rich sequence, especially in the consensus of GCC RCC AUG. And this sequence interacts with EIF1 factor. And remember that we said that it helps in the positioning of the mRNA, and that's where it comes into play. And this sequence here is the Kozak sequence. And now there's a small catch in this recognition of mRNA. Note that 40S subunit contains the 18S ribosomal RNA. But instead of ribosomal RNA, it is the initiation factor which helps in the recognition of the Kozak sequence. This is very different from prokaryotes, where we saw that the 16S ribosomal RNA was involved in the mRNA binding as well as the positioning of the start codon at the P site. Here in eukaryotes, the mRNA binding and positioning is initiated by initiation factors. So the 18S ribosomal RNA here is primarily involved in the structural configuration of the 40S subunit. I hope this is clear. Alright, let's move on. So when this pairing between the mRNA and the tRNA is established at the P site, the 40S subunit undergoes a conformational change at the mRNA entry channel, and it goes into a compact state, and we call this state a closed state. So this entire complex moves to a 43S closed complex. This complex is also called 48S initiation complex. Once this complex is established, the eukaryotic initiation factor 5 causes the GTP in the EIF2 to break a phosphate bond, which then as a result causes the EIF2 to form a complex with the GDP. And this complex loses its affinity towards the ribosome subunit, and as a result, it is released. The EIF1 and EIF2 interact closely, so actually both of them get released. And now this causes the EIF3, the scaffold protein, to release as well. And once the scaffold protein is out, nothing really stays on the ribosome. So all initiation factors, except for EIF1A, which is at the A site, are released. And if we draw out the resulting picture of the 40S subunit, you will find that eukaryotic initiation factor 1A is the only one which is present, which is at the A site. And the initiator tRNA with its methionine is attached at the P site. And the mRNA is still looped as the EIF4E and 4G are still interacting independently of the ribosome subunit. So to make this explicit, the A site is blocked by eukaryotic initiation factor 1A, the P site is blocked by the tRNA, and the E site is empty. Remember that we said that the EIF1 prevents the 60S subunit from joining. Well, now we don't have the EIF1 present anymore, so the 60S subunit can happily bind to the 40S subunit. But there's a small catch. The 60S subunit needs help. It cannot just bind by itself. So another initiation factor, EIF5B, which is in complex with a GTP, binds to 60S and delivers it to the 40S subunit. Specifically, the EIF5B interacts with EIF1A and correctly positions the 60S subunit. And when both of these subunits are correctly positioned, the GTP in the EIF5B is reduced to a GDP. And this causes this 5B factor to lose affinity and it gets kicked out. But when it is being released, it takes the initiation factor 1A with itself. And now, finally, we are left with 40S and 60S subunits that are still attached together with the initiator tRNA paired at the start codon at the P site. And the mRNA is looped because of this interaction between EIF4E and 4G. The E site and the A site are both empty. And this complex is called the ADS initiation complex, which is ready for the next stage, that is the elongation. And this wraps up our discussion for this particular video.